Hi guys, in this video we're going to talk a little bit more on vision. If you haven't already watched the 101, I've sort of done an initial video which I recommend, which talks a little bit about the structure of the eyeball, which will probably make a lot more sense if you watch that first. Okay, so basically we talked a little bit about how the eye is really a tool. A lot of the accessory structures of the eye, like the cornea, as well as the different humors, are responsible for refraction. Really, they're taking light that's coming into the eye like this and trying to get it to one point on the retina so it can be sent to the brain and understood. Now, the thing is with this is that we need to understand what the major surfaces are that allow that sort of refraction to happen. So those accessory structures, the first to understand is the cornea, which is, we talked a little bit about that strong outer layer called the sclera. It's sort of the outside area that sits, so that protective layer at the front is a translucent area at the front called the cornea. That is the largest point of refraction. So you may get a question on the quiz about like the largest point where the most refraction happens, and that will typically happen at the cornea. That will also happen, you also have in front of the lens, you have this aqueous humor sort of liquid that sort of sits here, right? And then let's call this the aqueous humor. And we also know that this area here is quite a like a water-like substance. It's very watery, and going through that, you're going to have further refraction. And then obviously behind that we have the lens, and we also have the pupil here. So what tends to happen is the refraction will come in from all of these different directions, and then it will sort of centerize at a core point on the retina. Now this, like we talked about previously, is an example of a convex lens. So you may also get a question, what kind of lens is the eye? And it is a convex lens. And it's important to understand that because we'll talk a little bit about how you can correct issues in vision later in this video. So one of the most important things to understand is that the image that is projected here, which I should draw that actually hitting the back. It doesn't, in normal vision, it won't, in vision areas, it may hit the sort of the point of where it sort of condenses. It could be here or here or here, but in healthy vision, it's hitting the back of the retina. So it's important to understand that the vision that's produced here is upside down, okay? And the brain and the central nervous system is what is sort of translating that the right way, which we'll talk about in later videos. So what can go wrong in this process? Well, if you've ever had anyone who wears glasses like me, they may have something called myopia, which is also commonly known as short-sightedness. Okay, and what happens in this case is there's a bunch of theories about how short-sightedness sort of happens, but what is commonly thought is that it is because the eyeball, so if you imagine a normal eyeball, which you've talked about, it goes through all the refraction processes, and the refraction ends up here. Okay, great, perfect vision. But what happens if we grow the eyeball? And this was completely freaky to me when I first heard about the idea of eyeballs growing. It was not really something I thought continued to grow as we get older, but it does. So imagine if the eyeball was longer. Now what's going to happen if we have the same amount of refraction is it's going to come in here and hit this point instead of the retina at the back. And what ends up happening is it then shoots back out again, and you don't have clear vision and you don't have clear sight. So this is an example of myopia, okay? And in myopia, people can only see short distances. Now, when someone has myopia, the treatment is, and I should also say, you might be wondering, how do you actually get, longer eyeballs? Like, what actually happens? Well, there's two major things. first is genetics. Some populations are sort of more predisposed to this. A lot of Asian genes are sort of associated with this. And the second one is sort of the environmental and behavioral factors. So if you spend a lot of time in front of a computer, a lot of time in sort of that environment, your eyeballs literally grow. And that sort of like when people say don't sit too close to the television, your eyeballs will fall out, etc. It isn't exactly true, but it is sort of true that they do grow. So what happens is that basically it's a problem of too much refraction. It's not refracting into the right place. And the way to fix this is we put another convex lens, we put another lens out the front here, which sits here, a convex spherical lens, and that overcorrects, and then it will end up in the right spot. Okay, so that is the correct approach to focusing light. You basically will make sure that you correct it with the glasses, and that's what an optometrist would do. Now let's talk a little bit about long-sightedness. This is also called Hypermetropia. So the interesting thing about hypermetropia is that it's the exact opposite of myopia, which is the short-sightedness. And someone in this situation is going to be able to focus on the background, but not be able to see things up close. And like we talked about with myopia, where the eyeball is usually too long, in this situation, the eyeball is too short. So what you can typically see is the light will come in like this, and it will sort of come through, and you think it's going to sort of focus, it should be focused here perhaps, but instead it focuses behind the retina. And it can't go that far because the retina is there, but you don't get a clear image at this point. So the eye instead focuses on processing the background, or the CNS processes the background. So in this case, you use a convex lens to converge it before it comes in. The other example of a issue that can happen in the eye is astigmatism, and this is basically what happens if you imagine an eye. We already talked about the cornea, which is that protective layer out the front here. I'll just draw this in a different color so it's more clear. Now the thing is that in reality, none of us will have a smooth cornea. It doesn't really happen in nature. So what happens is the light comes in, and most of it, that's great. It gets sort of refracted like we want, but some will get shot off on different angles because it's not exactly straight. And then what that ends up happening with astigmatism is you have light focused on different parts of the retina, and it has impact on vision because of that. And that is something that needs to be treated as well. Okay, so let's focus now on accommodation. So just to sum up, the three different vision issues we looked at were, first of all, myopia, where the eyeball is too short and you can only focus on the things that are very close to you, and that's because the refraction point is before the retina and it sort of spreads out very widely. Then we also talked about hypermetropia, which is also known as long-sightedness, and people can see far away but they can't see close. And in that situation, people focus on the background. And we also focus on the third thing, which is astigmatism, which is the light rays from different planes focus on different parts of the retina. So let's talk a little bit accommodation. What is accommodation? Well, accommodation is when you look at something, this is our eye, what happens when you get very close versus when you look at something very far away? And you can you can do this experiment, ask somebody to look far away and look close. And what you'll tend to notice are a few things. So the first thing is that looking at things very, very, very close, like let's say for instance, you put a pen right in front of your eyes. So you put a pen here and your eye is there. What's going to happen? Well, what's going to happen is that your pupils, the black part of your eye, are going to enlarge. okay? Because they want to get as much light in as possible. Sorry, so not going too large. Sorry, I said that wrong. They're going to shrink because it's so close. There's enough of that light, it's really close. The additional thing is that your eyes are going to be drawn closer in together. And what's also going to happen is that when things are very close, the lens here is going to become spherical. So instead of the lens looking like very relaxed, like it might like that in normal vision, that it's also going to look a lot like this. And that is to refract things at a different angle because it's closer, and it makes sense. If something's closer, it's going to need a different refraction level than if something's further away. So what you need to know is that when things get closer, the lens becomes more spherical and it has a greater refractory power, so it becomes greater. And this is controlled by the ciliary muscles. Remember we talked about in the first video, those ciliary muscles, they're going to contract and that's going to pull it back and it's going to create that shape. Okay, so just to sum up, what's going to happen? First of all, you're going to have a change in the shape of the lens. The lens is going to change, and you're also going to have a different shape of the pupil, which is controlled by the iris. So those two things are going to change. It's going to become, the lens here is going to become more spherical, and the pupil is going to become smaller. Okay. Now, what happens when we look at something far away? Got a building over here and we're here. What is our eyes going to see? And what are our eyes going to react to? Well, it's a little bit different this time. Basically, the first thing is the lens is going to be pulled flat. Okay, so it doesn't need to be as powerful because the light rays aren't as close. The light rays are traveling all the way from the object. So they're going to be, the lens is going to be flattened. Okay, lens flattened. And what is also going to happen is that the aqueous humor, that area is also going to be extended because what ends up happening is as we flatten this, there becomes a more space here for that liquid. Remember we talked about that liquid that's involved, the aqueous humor, which also helps affect the refraction that is going to occur. And the pupil, which is controlled by the iris, is going to become what? Well, it makes sense that if the pupil is going to become smaller when things are closer, it's going to become larger when things are further away. So that is basically how accommodation works. Now, one really important thing to think about here is, and quite often is a question, is what is the impact on age? Now, usually what I've found is that in the research and the text is that 50 years is usually the point where the sort of plateaus. And up until 50 years, this refractory process progressively gets worse. So, when you're born, your near point, which is the point in which this process starts to happen when you move from having a flattened lens to a spherical lens, when your irises go from large to small, that near point progressively gets further and further away. If I'm young, I'll be able to, get things to up to they're really, really close before I get this reaction. But when I'm older, it's because it takes longer and longer, and it's a further away and it starts to happen. And why is that? Well, one of the reasons that we think this is because these ciliary bodies tend to become stiffer. And remember, they're controlling that lens flattening. So if these ciliary bodies are becoming stiffer and more difficult to move in time, what ends up happening is that accommodation process becomes larger. Cool. So that's what accommodation is. Now, let's talk a little bit about the process of the iris. Now, I've hinted at a few points so far that the iris is involved in the regulation of how much light enters. Okay, so in a really bright room, how big do you think the iris is going to be? How big is the pupil going to be, I should say? The answer is, in a really, really bright room, the pupil is going to be very, very small. In a very, very dark room, it's going to be very, very light. And the eye adapts based on the amount of light that's required. Now, this is controlled by the iris. The muscles around it are the iris. Now, when you see people with blue eyes or green eyes or whatever, that is their iris. I don't think you've probably seen anyone with purple eyes, but you'll have to bear with me as I make this presentation. So that is really what the iris is doing. Now, the interesting thing is the other thing that the iris will do is reduce something called spherical aberration. Now, what spherical aberration is basically when the outer part of the lens, so maybe these parts, they're actually not they're not as effective as the internal parts because there's too much refraction, or maybe it's not, there's too much light. So what it does is it closes it to become more focused. And that's why when you want to get closer to something, when something is closer, your iris, the pupil will become closed by the iris to prevent light from entering into to the eye and really improving the quality of the image. Because another thing to really remember as well is the visual acuity is greatest in the middle. And don't worry, we're going to talk a little bit about why that is, and that's got to do with receptor fields and things like that. But all you really need to know at this point is that the iris opens and closes. It controls the size of the pupil, which controls how much light goes through the lens. Okay, great. Now, the thing is that let's talk a little bit about, let's deep dive now into the retina. So we're going all the way to the back of the eye. Now, we talked about the back of the eye, and this retina level is the innermost level. So obviously we've got these two other layers here, which we're not going to talk about in this part of the video. So at the retina, there is actually a very highly structured process of how these cells are laid out. And it is a little bit counterintuitive. So, if I asked you, okay, well, if you were to tell me where you think the photoreceptors would be here, where would you say? And I think most people would say the photoreceptors would be on the outside because that's where the light is hitting. But that is not the case. The photoreceptors are actually in the furthest back distance. They're the last cells that you see here before the end of the retina. That's where you'll find the photoreceptors. Now, there's two different kinds of photoreceptors, and we'll talk a little bit about those as well. There's rods and there's cones. Now, rods, we often associate these with like dark vision, and we'll talk a little bit why. It's due to their adaptation rate, as well as cones, which are often used in the daylight, and are often used they're often used as well in color, and very selective for color. They have high visual acuity, which means they can see a lot of detail. So a couple of buzzwords here that are really important to remember is that all vision is G protein linked. So I'm not going to go too much into what a G protein is, but it's important to understand that the way this message is sent from the outside into the brain and to other parts, and from the receptors through to the other neurons, that process happens via a cascade of enzymes and proteins. So it's not just one thing doing one thing. It'll basically be a GDPR, which is a G protein associate cellular receptor, and basically this is called rhodopsin. Okay, rhodopsin, and that sort of sits on the cell membrane like this, of some of those different receptor cells, and then basically the rhodopsin activates the G protein, which is called transduction. Ductin, just a pretty good name if you're going to call something a G protein. Then, basically, the process then is PDE, which is the enzyme, PDE. And if you want to know exactly what it is, it's called phosphodiesterase. Phosphodiesterase is responsible for producing the process of cyclic GMP. Cyclic GMP is really what opens that the level of this opens and closes the ion channels. So basically in the dark, there's lots of cGMP. So basically, there are in the dark, there's going to be lots of channels that are open, and hence there's going to be lots of there's going to be lots of depolarization and action potentials, which go on into the nerves. Then, in light, basically, light activates a breakdown of the cGMP, which causes a closing of the ion channels. So the ion channels of the cells close ion channels. Then, once the ion channels close, you'll basically have hyperpolarization. The cell's membrane potential will go down. Hyperpolarization will occur, and the resting membrane potential will go down. The advantage of this process is primarily amplification. Amplification basically means, the question you might ask is, okay, well, why do we have to have such a complex system, which is PDE, cyclic GMP, opening and closing ion channels? Why can't we just make it super simple, maybe jump directly to this, opening and closing of ion channels? But really, that's not the best way to do it, because amplification is important, where you can take one photon of light and literally have hundreds of thousands of sodium channels, hundreds of thousands of sodium ions moving in and out of cells, depolarizing, hyperpolarizing cells, which is used for signaling. And so that's really the main advantage of that process. So that's pretty much it. Thank you for paying attention. In the next video, we'll talk a little bit about light adaptation, the differences between cones and rods, and some things like that. Thanks for paying attention. If you have any questions, please ask them in the comments.